everybody. Welcome back to the fourth week of the Boulder Summer School. Some of you already know me. I was here at the end of last week, but I haven't introduced myself. My name is Eric Dufresne. I'm a professor at Cornell University, and I'm going to be the lead organizer this week. So if you have any issues with respect to the school, just let me know, and I'll try to take care of it for you. So this week we have an exciting series of lectures. We're going to sort of move the biology up to sort of the tissue scale. And then we're also going to move into new areas, thinking beyond mechanics and start thinking about information and computation. So this week's lecturers are our first lecturer, Young-On Mao from the University College London, who will be speaking to you today about tissue mechanics. And then we have Andrea Liu from the University of Pennsylvania sitting up front. She'll be speaking later today. Doug Durian, also from the University of Pennsylvania. I think your first lecture is on Wednesday. Yep. And then my colleague Ty Cohen should be here soon. He'll be speaking this afternoon. And we'll also have Vincenzo Vitelli joining us at the end of the week. So without further ado, we'll just dive right in and we'll hear from Professor Mao.
Great.
Thank you very much.
Okay, thank you. Can you hear me, by the way, at the back? Yeah, because I'm sort of slightly losing my voice. I'm not sure if it's the altitude or the jet lag or the heat, but hopefully I don't have to shout. So if you can't hear me, I can turn up the volume. Okay, wonderful, good. So yeah, it's a pleasure to be here. And I think I'm kind of the bridging set of lectures between sort of tissue mechanics and then the next series on sort of the active matter, morphogenesis or whatever the next topics are. So I think I was meant to be last week, but basically due to many reasons, I couldn't make it. So I'm here this week instead. And just to really introduce the GAN and set the scene a bit. So and also just to thank everyone before I forget at the end of tomorrow. So basically this is the kind of the current team pretty much, and also the past and current members. And essentially we're an interdisciplinary lab. I'm actually trained as a biologist, but in the lab we have physicists, computer scientists, engineers, and biologists, geneticists. So we work together really as a team to answer some of the questions I'll talk to you about today. And we'll try to, I've heard that you're a bit tired, so let's try to, and it's also 9 a.m. on a Monday morning, so we'll try to keep it relatively simple for the first, half an hour or so, and then we'll sort of delve into some problems. And also, I think you a lot of equations. You're all probably much better than me at solving these equations. So my job is not to, teach you how to solve equations, but really to do and how to actually bridge these experiments with the theory, with the models that you've hopefully learned about already in the last few weeks. And really how, as biologists or biophysicists, how do we really combine all that together into, driving some of the kind of the current research that we're doing in the lab. And so just to set the scene, so essentially my lab, what we're interested in is understanding how these beautiful patterns in nature are formed, right? So this terrible photo was taken by me on my really rubbish iPhone from like 10 years ago. And the reason for that is because I went to this museum with my kids and I saw this display and I wrote down the top, this key sentence, which really made me kind of go, hey, look, this is exactly what I work on. And this little museum in my hometown in Glasgow was basically saying the same thing, which is that the genetic instructions and building blocks used by organisms are the same. Yet we have this beautiful diversity, right? Whether it's animal patterns and shapes or plant shapes and so on. So how do you actually get this awesomely beautiful diversity? And you've heard lots of talks in the past weeks about this, but that's really what's been kind of driving my lab's interest in the last 10 years or so since we started the lab. And of course, as we all know, it's all in our genes. There's absolutely no denying that, as much as all you physicists like to think it's all about physics and physical principles. But I think we also have to admit that genes are, of course, essential, right? And there's no denying, it's pretty obvious, right? Our kids look like us and we look like our parents don't. But I guess the fascinating thing is how do these genes give rise to the beautiful range of sizes and shapes that we see in nature? And I think that's still the biggest question that at least is fascinating me every day in my group, and I'm sure many of you here. And so, again, I hate these diagrams. Lots of biologists use these diagrams. So, various omics approaches, right? Whether it's proteomics, genomics, RNA-seq, this could be genes, it could be proteins, it could be signaling networks, it could be, whatever you call it, these kind of mothball diagrams. And of course, probably most of the biologists that we work with are quite obsessed with these kind of diagrams and kind of configurations. And yes, they're absolutely essential and useful for much of our understanding. But I still find this very unsatisfying, probably the same with all of you, right? How does this actually tell us how do cells move, right? How do they divide? How do they grow? How do our organs and tissues morph, right? And of course, maybe as no surprise, we are therefore interested in really the mechanics, these physical forces, and how are they generated as a result of the genes? And this mechanics really means mechanical forces as well as material properties, the mechanical properties, right? So these are all set, of course, by the genes, but also collectively, this can give rise to shape of the structure, but also there's constant feedback interplay between all of this, right? So your shape of the cells of the organ can link back to affect how genes are expressed, gene expression, and so on and so forth. So this is, I think, again, you probably don't need convincing by me now that these are all interlinked. And it's therefore, how do we untangle this, or how do we understand this linkage? That's the key to understanding the whole system. And so tomorrow I will also talk a little bit about these cell intrinsic factors, which my lab is starting to be more and more interested in. So a lot of this, of course, you can think of it as cell intrinsic forces. How do genes regulate the internal mechanical forces? How do they generate forces and so on? But also, in fact, we're constantly affected by things around us, whether it's the external matrix or, in fact, the internal environment. So we'll also hint a little bit about that tomorrow. Okay. So overall, again, just to introduce kind of the theme of the two lectures that I'll give and the logic behind it, essentially, it's sort of split into three conceptual parts. Okay, the first part is really about development and morphogenesis. But then the second part is about homeostasis. So how do we actually stay in shape? This kind of concept that, in fact, for most of our lives, we have to try to stay in optimal shape to continue functioning. And then finally, if we have time, we'll talk about tissue repair and regeneration. So essentially, if you are damaged, how do we get back into shape? And obviously, it's all kind of linked to organ and tissue shape and so on. Okay, so just to give you kind of an overview of the structure of the two lectures. So today we'll talk about growth and morphogenesis. So there will be some very basic background, because I don't know exactly all of your backgrounds, but this is just to give you a very quick overview, mostly really linking some of the key biological experiments that the field has seen over the last 20 years or so, and how that's being really linked to the idea of growth and morphogenesis and how mechanics is conceptually important. So just so that we're all kind of on the same starting point. And then we'll look at a case study of using vertex modeling of tissue shape. So I think you've, again, Lisa Manning introduced vertex models, so that's good. That means we don't have to go into the detail, although I'm sure some of you have used it anyway in the past. But then it's really about how have we used this to understand, for example, this concept of shape control. So I'll use a case study to kind of use it as an example to show you how we've linked genetics, experiments, and the model, and this kind of back and forth. How do we actually use that to understand the problem we had? Okay, and then day 2 tomorrow, as I said, we'll go into the concept of homeostasis and repair, and I wanted to show you some. So today is more kind of background and old work, let's put it that way. And tomorrow will be much more about the latest research that I want to share with you that's happening in my lab, which I'm super excited about. So I think it's also really important for you to learn about some of the latest unpublished stuff that we're doing and the new tools and methods that we're developing to answer some of these questions, all right? And if we have time, we'll go into, again, a very small case study about modeling of tissue repair, but let's see how it goes, and then we'll kind of play by ears, okay? And there are some exercises which I think I've shared already. It's probably uploaded already if you want to take a look, but I'll introduce the problem later today at the end of today's lecture, so that it's not completely random as you read it on the slides. Okay, so is that kind of reasonably clear, the layout? Again, as always, interrupt, please ask questions, and I can always tangent out and discuss whatever you want me to discuss. And so obviously there is a structure, but again, feel free to interrupt and we can just fluidize the lecture, all right? Good. And also, if anything is like, oh, you've heard this 10 times already, let me know. We can skip it, because again, I tried to look at some of the videos, but obviously I didn't look at all whatever 20 lectures that's uploaded on the internet right now. So again, feel free to just, say, oh, I've heard this, skip, and I can skip. Okay, so good. Right, let's start. So, like I said, first half an hour, just really some basic intro. But again, just to try to step back into basic principles, right? If we want to talk about tissue growth and tissue shaping, what are the very basic processes that need to happen at the cellular level? And the way I think of it is, you need to have growth, right? Cells need to grow, need to divide, they need to increase in mass and material, no denying that. But as part of that, you also need to have cells dividing. Most of the time, we're not a single cell that's growing infinitely. Most of the time, we are single cells growing and dividing and dividing and growing, right? So cell division is another super key process that's, linked to growth and morphogenesis, right? But also then if you start talking about morphogenesis or shaping of an organ or a tissue, then you also have cell shape changes. Well, that's at least one thing that potentially could also affect the shape of the emergent organ or tissue, right? Simultaneously, I think some of you, I think this was touched upon in some of the lectures. So this idea of cell intercalations or cell neighborhood exchanges, again, in this room, if we all started to move around, that's another way to keep our individual shapes, but change the collective shape of the tissue, right? And finally, in 3D, again, none of us are flat epithelia. We do have 3D morphology, and so this 3D tissue sculpting is another really key important part of how we control our organ shape, right? So of course, this is sort of conceptual how I've split, the process up. And of course, often this happens in parallel. They're not, linked to sort of sequential events. They happen in parallel, and it's really about trying to untangle the importance of each process in different systems, right? And as I said, these are the cellular processes, and many, of course, biologists have been looking at these from a pure, biology point of view. But I think it's become pretty clear in the last, 10, 20 years or so that physical mechanical forces really can play a huge role in each of these processes, and it's therefore linking all of that. That's essential to understanding these cellular processes. And so let's sort of focus on first one, which is the control growth rates, or at least how mechanics links into growth rates. And again, I'm not sure how much of this is being mentioned, but clearly, growth rates are regulated by what's called the cell cycle, right? Cell cycle, there's different steps. You have growth phases, you have replication phases, you have mitosis, and so on. So these have been studied by scientists for, 50 years or so now, maybe more, right? And so these steps are crucial to driving the whole process of cell and tissue growth, okay? And I won't go into the details. There's textbooks and hundreds and thousands of papers written about these. But the key thing is many of these growth factors or biochemical signaling pathways that are known to feed into each parts of the cell cycle and drive the cell cycle, many of them are also linked to mechanics, right? Or are mechanosensors or mechano-responsive factors. So for example, I'm sure you've all heard of the Hippo, YAP, TAZ, Yorkie pathway. No, actually no. Okay, great. This is good. So if I say, oh, maybe you've all heard of it, please tell me no. So who has heard of YAP, Yorkie, TAZ? Okay. Oh, actually, okay, good. This is good. That's good. So I was worried about, just telling you everything you already know, but okay, good. So this is important then. So, you've heard about all these beautiful equations and theories, but how does all of that link to biology, right? And this is one of many, many links, right? So this is hopefully interesting for you guys to understand a little bit about. If not, we can skip it. If yes, let's go over it very quickly. Okay, so as I said, yes, forces, how do you actually link it to growth and all these material properties and things we've talked about? And one way is to actually make sure that certain molecules that affect, for example, cell growth are, of course, sensing these physical properties of tissues, right? Otherwise, how do you have this crosstalk? How do you link the physics with the biology, right? And so as one example, there are many now, but I think this is probably the most famous or indeed critical molecule, which is called YAP and TAZ in mammals, but it was first discovered in flies. Hence, as always, we like to complicate things as biologists. There's like 3 ways to basically say the same thing. So let's call it YAP during these series of lectures. And what is known about this pathway is that normally it's essentially a gene transcription factor which controls cell size and cell number, okay? So at the simplest level, what happens is that this Yorkie, when it goes into the nucleus, it drives the cell cycle, so it makes cells grow faster, it makes cells divide more. That's all you need to know, okay? So very simply, if it's in the nucleus, more growth. That's, I think, for physicists, that's probably all we need to know. And so we're just trying to simplify the problem. And upstream, there's, again, hundreds of molecules upstream. But what's important here is, for example, the mechanics can be an upstream signal for the transduction of this key molecule, okay? And how does that work? So essentially, again, about 10 years ago, what people found is that, incredibly, if you grow cells on different stiffness substrates, so if the cells are sensing different mechanical environments, then this key molecule, your key, will either go into the nucleus, where it turns on more growth, if it's on, for example, very stiff substrate, okay? So essentially, again, the simple picture is that if cells are growing on stiff substrates, this causes them to spread their cell shape into more kind of fried egg shapes. And as a result, this causes your key YAPTAZ to transduce and move into the nucleus, which means that you have more growth, the cell cycle, and so on. And the opposite was also true, is that if you have cells growing on very soft environments, soft gel environments, then this Yorkie factor moves into the cytoplasm. And as a result, genes that would normally, for example, cause cells to grow and divide more, those are turned down, and you have more of a growth at rest, okay? And the reason why this was so exciting was because many years before this, people had shown that if you grow stem cells on stiffness substrates of different stiffnesses, these stem cells will actually differentiate into different cell types. This was kind of in my opinion, probably the most exciting kind of work which started the whole mechanobiology field, is that cells actually can sense the mechanical environment and behave differently depending on their mechanical environment, right? And for example, for stem cells, that means they can grow into different cell types. They will differentiate into different cell types and adopt different cell shapes. But exactly how do they do that at the molecular level? This concept of mechanotransduction was kind of a mystery to many biologists and many physicists. And how do you really link, the biology and the physics together? So anyway, so this is one example. And I guess basically, if you the PubMed literature now, pretty much I would say every day or every week at least, there's a new paper claiming, another signaling molecule is mechanosensitive and so on and so forth. But this, I think, is just one concept, at least, of how your mechano environment can really affect, growth and cell, cell growth, at least, and differentiation in this case. Okay, and so just to therefore show you different ways in which biologists can kind of control cell shape. And so there are techniques called micropatterning, where you can force the cells to spread across different surfaces or be very confined and end up looking, very, very sort of rounded and small. And as a result of this, for example, in this case, they spread the cells out. You can see Yorkie or YAP goes into the nucleus, whereas the opposite is the case if you force them to grow in a very confounded, kind of confined space. Yes, question? Yeah. Yes. Many of these are comes, but it's true. It is kind of different. Also depends on how you grow them. So, for example, in this 2D setting, it seems to be the case. But what people have found that in, for example, 3D organoids, the opposite seems to be the case. So people are still kind of trying to understand. So a lot of these experiments and a lot of the lectures I'm focusing on are all still in a more of a 2D setting. So, for example, in this case, it's 2D cells growing on top of a gel, right? And they found this, but in fact, it's been shown that recently, if you grow this, for example, as a 3D ball or a spheroid or organoid, the opposite seems to be the case. And there's, hand wavy reasons for why and so on. But again, I think most of the time it's really linked to the cell shape, but also the cytoskeletal tension and actomyosin that's transducing the signals and how they then link into the Yorkie transduction. Yeah, exactly. So it is a little bit context dependent, cell type dependent, and how you grow it and the sensings. But again, I think all I want to say is that certain genes for sure are sensitive to the mechanics, and as a result, they will, switch on different things. Yeah. Okay, good. So anyway, so there are techniques to kind of change shape. And as a result, by changing cell shape, you change these key growth factors, okay? And there are other techniques, for example, they can grow these cells on these kind of rigid or soft pillars. And as a result, the cells are able to contract and change shape. And again, they are affecting the kind of the localization of these key factors, okay? But also another very kind of mechanical process that is crucial is mitosis, right? So again, I'm sure, have any of you actually looked at a cell undergoing mitosis down a microscope? No? Yeah, some of you, yes, some of you, most probably no. So if you do have the chance, I would encourage you. To me, it's still the most beautiful process, on Earth. But also you'll see it's a very mechanical process because cells really have to change shape dramatically to divide, if you've seen them, right? And most of the time, most cells is the fact that, there will be a resting shape. It could be this kind of flat pancake shape, but during mitosis, they have to most of the time round up, change. For example, in this case, this cell becomes pretty much a sphere. And the reason for this is they really need a huge increase in volume of space to actually assemble the mitotic apparatus to undergo cell division, okay, which is actually a very complicated but beautiful process. But you can see here, it's very mechanical. They have to change shape dramatically to enable this, okay? And so again, most of us here, our cells are all quite compact. How do you actually undergo cell division in a confined, compact space? And how do you enable these dramatic shape changes, right? And so people have done these, again, beautiful experiments where they try to squish cells between, let's say, a soft gel or a hard gel. And if the gel is quite soft, then you can see these cells generate huge forces, which enables them to round up. And if it's a soft gel, these soft gels will be kind of basically pushed out of the way. And so the cells can round up and undergo perfect, mitosis. However, what we're shown here is that if you have a hard gel squashing these cells, the forces that they generate aren't sufficient to push, this hard gel out of the way. And so these cells are forced to kind of divide in this very confined, constricted volume. And as a result, you have basically problems during mitosis. For example, chromosomes don't segregate. And obviously that's a problem because during development, you're trying to grow your cells, divide your cells to make sure there are no errors, right? And if this is an error, then of course there's many developmental defects, diseases, and so forth. So again, just to show that this critical process in life, which is mitosis, which enables you to increase the number of cells, this is an incredibly mechanical process. Yes, yeah. Yes, people have. So again, there's a range. So for example, I think it was Tony Hyman measured some of these numbers. And again, obviously depending on cell type, but there is exactly like a range of, and that can be done, absolutely. So people have put, for example, AFM cantilevers to as a way to sense how much force is generated. So yeah, exactly, yes. But again, linking back to everything you've learned so far, right? So if all these physical parameters we've been talking about in the last few weeks, how do they actually feed into, for example, these critical cell processes? What are these, processes that are, go on. So yeah.
Those bigger cells we get
yeah, so exactly. So these ones, so not multinucleated, but sometimes, for example, the chromosomes, sometimes they divide into 3 cells. So there's chromosome missegregation. So again, normally you should have, 2 perfect daughter cells and so on, right? But as a result of this, you can already kind of see in this picture, I didn't show you the full images, but what they found is sometimes you get 3, which obviously is wrong, problematic. And so yes, you can have basically the DNA content goes wrong. Yeah, exactly. And some of them will die as a result, but then that would be good. But sometimes obviously that's then inherited and that would lead to a huge problem for the animal or the plant. Yeah. All right. Okay. So again, just to quickly kind of link that. But a lot of these, of course, were just in vitro single cell experiments, right? Just to kind of relay the concept. But again, most of us, we exist as multicellular organisms, right? And in a multicellular system, this becomes even more complicated. So this was a paper that my lab published a few years ago, where it was kind of the same idea. How do we actually regulate this mitosis in a multicellular, already very crowded system? And the idea here is that basically what happens is during mitosis, the cells actually, the nucleus have to migrate up into the apical surface, undergo this rounding in this apical region where the cells have a bit more space, and then they will undergo division and then move down again. But if you have a crowded tissue environment, so what we did is we designed a system where we could actually stretch or compress the whole tissue. And as a result, you can kind of generate delays in mitosis, you can generate problems in mitosis, again, just to relay the effect that external mechanical forces or how the cells really experience these forces will affect how efficient that mitosis is, and as a result, obviously errors and so on. So, okay. Yeah, so again, I will share these slides online later. So there are papers and stuff. I won't go into details during the talk, but the references are there if you want to look it up and so on. Okay. And yes, another very important concept is cell death, right? So again, linking to growth after is always a balance between new material and loss of existing material. And cell death, for example, cell extrusions. So you probably heard about these transitions, intercalations, but also cell extrusions. These are also incredibly mechanosensitive. So again, mechanics can sort of squeeze epithelium and cause death and cells to extrude, okay? So these are sort of a quick overview of the different ways in which growth rates can be regulated and how mechanics can feed into that. But also division orientations. So as I said, imagine if you're trying to sculpt or shape an organism or a tissue. Of course, you could grow in a kind of and divide in a very random way, and you'll probably get a sphere or circle, right? But if you are dividing already in an oriented way, then this orientation of cell divisions will already kind of guide the axis of elongation. So we'll talk about this in the case study later. So division orientation is another way to control the shape of organisms. And how do you actually control cell divisions? So essentially very simple. It seems to be linked to cell shape. And again, this was noticed, as you can see, hundreds of years ago, right? So basically, essentially giving the idea that if you have a long cell, pretty much always it will divide along the long axis so that you have two more isotropic shaped cells. And this has been kind of a rule that's being observed across, really many, many different organisms. And so the idea there is, well, how can you then control cell shape? If you control cell shape, then you can control division orientation, and then you can control the shape of the tissue, right? And so again, some beautiful experiments where scientists can, force cells to grow in different patterns, adopt different shapes. And again, they might not be so obvious. The shapes are anisotropic, but as a result, you have different stress axes, and these can actually somehow direct the cells to divide in different orientations. So these are, again, tools that scientists have used to try to understand what controls mitosis directionality, okay? And it's very much linked to the mechanical environment. And also people have done experiments, including ourselves, where you can force cells to become more elongated, change the cell shape. You can squash them, stretch them. Pretty much always, you will be able to change the direction of cell division just by changing the shape of the mother cell. So again, this was pretty kind of cool, I think, for sort of people considering morphogenesis and so on, right? And also, for example, in the models we'll talk about later, how do you link this very key concept of cell shape to as a parameter into your models to look at the emergent behavior of organ shape, right? So that's kind of one thing. So we'll focus a bit more on that later in the case study. So that was a very quick intro here. But also, as I said, just simply at the single cell shape level. So, again, we could be all sitting here, but if we started to just, let's say we all held our hands, how do we collectively stretch out? And one simple way is just if we just stretch our arms out, right? And if we change the individual cell shapes, the whole room or the whole collective would also change shape. So again, a single cell has many ways to change shapes, but if you are living as a multicellular system, your neighbors will, of course, impact how your shape is affected, right? And so these are kind of key biology concepts in, for example, the vertex models that you heard about last few days. And how are these parameters in the vertex models determined? They are actually based on biological observations, not just kind of, randomly invented by scientists. So for example, if you're this middle cell, right? In terms of, how you link to your neighbors. You could increase your adhesions, so you could extend these yellow junctions with your neighbors by increasing your adhesions. So remember, in the vertex model, this would be the line tension term, maybe a low value, maybe a negative value, right? This extends. Did Lisa go into this? I remember she did go into some of this, right? Okay, we'll revisit that later. But anyway, the idea is that if you're sticking much more to your neighbors, then you tend to extend the junctions or the connections with the neighbors, and that can change your cell shape. Or, for example, your neighbors could be squashing you more, could be, stretching you more. So as this kind of orange cell, you will also be non-autonomously affected by your neighbors because they are pushing you, they're forcing you, and stretching you and so on, okay? So all I want to say here is that in terms of thinking about cell shape, in the multicellular context, you have to think about cell intrinsic forces, but also cell non-intrinsic forces, these forces from the cell environment. And often this is easier to be studied if you have a model to kind of capture all of these dynamics, right? So for example, in this case, I don't know if anyone can tell me what they see is happening. So this is a simulation of a vertex model, 2D vertex model, where hopefully you can see cells are growing and dividing. So every time you see a kind of a balloon, it divides into two. That's a mitotic event, right? But then as I play this, can anyone tell me what's happening? Just whatever you see. Yeah. Exactly, brilliant. So cell shape here, you can see them already starting to elongate, right? They're changing shape, stretching circumferentially around this, okay? And do you have any hints? Can you sort of explain why that might be? What's going on to drive this? Yes and no. If anything, T1 probably releases this and actually dampens this effect more. But certainly you see lots of T1 transitions here. So let me play this again, right? So yes, there are T1 transitions happening. And you'll see actually quite a lot of them in this kind of corner. It's almost like the tissue is ripping apart, and obviously they can't rip conceptually in the model. So there's so here you see, lots of T1s here, right? So these are pushing in. And actually, if you look really closely, after every T1, the cells actually almost become less elongated because the T1 is releasing that stretching force and almost rescuing that stretch, right? So T1s are almost dampening that stretch. But again, maybe if I just play again, yes. Yes, absolutely, exactly. So beautiful, someone said the shapes, very clearly the emergent outcome is the cell shapes around the circumference, they're being stretched, they're elongated. But in fact, the reason for that is not because these cells somehow know they should be longer, or in fact, they're feeling this, what I call the non-autonomous pressure force from the middle. So because the middle are growing and dividing much more, you can probably notice that in fact, these outer cells, there's no division happening. So what we did was in this simulation, we only had growth and division in the middle. These outer cells, if you look really closely, none of them are dividing. So division is a way to release stresses, right? And if you don't release that stress, what happens? You stretch and elongate. And so this middle cell is actually, these middle cells, sorry, because they're growing fast, they're generating a pressure that's pushing out, right? And as a result, it's stretching these outer cells. And if I turned on divisions, they would divide and release that stress. T1 transitions would be another way. So we can also tune, for example, how much we allow these T1s to occur, and that would also set how much these are stretching. But essentially, it's this buildup of. So I guess the point I'm trying to make here is that you might think, oh, these cell shapes are all very intrinsically controlled, right? So scientists, in fact, spent ages trying to understand just conceptually, why are these cells stretched? Do they know they want to be longer? Yeah, go on.
So if your edge is under tension or
yes,
right? So you can release stresses like that.
Yes.
Does that like do something to the ones inside that are like dividing to release stress?
Yeah. So okay. So absolutely perfect. So we'll touch on that later. So this obviously was only a 2D model, right? So there's no third dimension for it to buckle, but absolutely. In reality, what happens is they would buckle, yes. And that is a way to release the stress. And as a result, then that allows the middles to keep growing. So this was introduced as a kind of concept of basically, many of you may have read the amazing papers by Hufnagel and Boris Schreiman, trying to link the idea that, this here initially going faster, maybe because of those growth factors I talked about earlier, let's say Yorkie is more in the middle. But then as a result of this growth, the forces experienced by the cells end up being different, and they will feed back into, for example, these growth factors. Or they could also just release these stretches by either dividing more, which again, people have shown. So I've done experiments where if we stretch the epithelium, you actually increase cell division rates, but also you can generate buckling. You can orient the divisions in different ways. All of these are ways in which this tissue can release these stresses, because if you don't release them, they could actually cause these middle cells to stop growing and pause, right? Which maybe is good sometimes, you want that, but maybe it's also bad if you want to kind of grow your organ, grow your tissue, right? So this already you see in this very simple simulation, super simple simulation, you can see lots of complicated emergent behaviors, right? And if then there are these feedbacks happening, you can see how this could actually be quite a complex system. And then if you introduce 3D, which actually we did in the lab, but which I probably won't have time to talk about, then it's another level of complexity. How do you, release these stresses? But yeah, absolutely. Okay. Yes.
Here, but how do you choose what is interior and what's exterior?
Yeah, no, this is the concept, right? This is just kind of a proof of principle. So I think we set like a hard barrier. We kind of had division rates here, and then boom, it goes down. But we also, in this paper, we also did, we tested different rules. So different gradients of growth rates. For example, it could be very slowly decelerating towards the edge, could be a massive step function, could be, so these are things you can play around. So, but however, they all kind of gave more or less the same conclusion, which is as long as you have a differential, you will have differential stresses building up. And as a result, you'll have different cell shapes emerging. But of course, that steepness of that gradient will determine exactly the final shapes and the stress buildup, right? Yeah. So exactly. But then ultimately is, whether you can measure these in whatever experimental system your biology collaborators are working on. Because again, I think the point is we can theorize as much as we want to, in these simulations, but do they actually play a role in whatever system biologically that people are looking at? That needs experimentation to test. And luckily there are ways. So what we did, in fact, and again, I digress, but it's good to have these discussions. In this paper, we actually, the issue here was, how do you generate these shapes? And so through the vertex models, we actually said that there's probably only one way, which is if you have this massive differential of growth. And that was quite controversial in the field, actually, because basically people thought the growth parameters were flat, so the cells are not growing differentially. But the modeling really kind of gave us a hint that this is really important mechanism. So then that encouraged biologists to go and remeasure some of these growth rates using newer techniques, newer methods, and actually people did measure a differential. So this is where I think theory has really helped guide, experiments and experimentalists, and has really kind of fed back into each other. Okay. So there'll be a reference for that paper at the end if you want to look into it. We can talk about it at lunchtime or whatever. Okay. So I guess the concept here is, again, you might see cell shapes changing, but in fact, the cause of that could be somewhere very far from that particular cell. And it could be these, other global forces, okay? And so, yes, then another way, I think you've heard a lot about this from Noah and from maybe Lisa Manning, these kind of T1 transitions, cell tissue fluidity, right? So again, you can change shape by, for example, just changing your neighbors. In this case, the cells haven't changed shape, but they've changed their configuration. And this is one way to elongate. And again, this was studied really in high detail. For example, the reasons why these T1s occur is because, for example, you have myosin 2, which is polarizing these forces, okay? And as a result, this will drive these oriented T1 transitions. So I'm sure you've sort of probably heard about this already. And we've shown that, again, for example, these can be regulated in different ways. So for example, if you were to stretch a tissue, in this case, a wing disc, then these milestone polarization, which people have shown in these cases, can actually themselves be regulated by mechanical forces. So this is, again, where the feedback becomes very complicated. So there are molecular signals that control the polarity, but this polarization itself is sensitive to the mechanics of the cells. So for example, if you stretch tissues, milestone will polarize, and this almost prevents further cell shape changes. So this polarization is a way to drive intercalation, but it can also be a way to prevent intercalation from happening. Okay, so this is, again, where I think in terms of when you think about your models and the context of the models and why, in different biological systems, your model can explain one scenario, but not another scenario. I think, the context is, of course, very important. All right. And the importance of these intercalations. So this idea of tissue fluidizing. Yes, go on. Yeah, sure. Yes, it's the same, it's exactly. So I prefer to use the word intercalation just because it's less jargony than, T1, T2s, and T3s and so on. But again, mainly because some papers don't use T1, T2, T3 the same way as other papers. So it's just less confusing. But I think most, maybe 90% of the theory papers have used T1 transitions as a way of describing intercalations. But yes, basically this neighborhood exchange. Okay, yes. Right. And again, so tomorrow, if we have time, we'll go a little bit into this paper where we kind of showed that, in fact, these intercalations or T1 transitions, what is the actual function of them, right? So they can be used, of course, to shape organs and so on, but also we showed that in terms of tissue repair, wound healing, what happens is that if you have a wound or a hole, then the cells around the wound actually increase the intercalation rates or become more fluid. And as a result, this can drive more efficient wound healing. So how we got to that conclusion, we can talk a little bit about it tomorrow if we have time, but essentially a really unique role for this kind of process of intercalations is to actually heal a wound in a much more efficient way. And the concept there, I guess, is, imagine you have an ice cube, you punch a hole or, carve out a hole in it. And the most efficient way to heal that hole would be if the ice cube melted, right? And so that's kind of how we've sort of conceptually shown that this is actually what happens in lots of tissues to give you much more efficient repair, but also much more what we call, basically that repair gives you less scarring, less kind of, less scarring, okay? So again, if we have time, we'll go into that. And then finally, so this very much links to the idea of 3D intercalations. And this is the question that you had earlier, right? So basically this, the simulation we had earlier was this, but only in 2D, right? So if you had, a sheet of kind of elastic, viscoelastic material, and if you were to stretch it, it would stretch in 2D for a while, but then if you keep going, it would buckle, right? And there's been lots of theories on this, and essentially what we've also shown is that this could be a way to actually generate these outer plane deformations, which is key, of course, for many organs, right? Whether it's the gut, the brain, the branching of lung epithelia and so on, right? So these are all essential biological processes that require this outer plane deformation, and how they're actually controlled, how are they generated? And so lots of work from physicists, biologists, so on. I'm sure you recognize some of the names here, different theories, different principles, whether it's compression in a confined space or differential growth of different Z layers, for example, in the brain, or simply just the top and bottom of the cells constricting differently. These can all generate these different outer plane deformations, right? But also what we showed, which was linked to that simulation I showed you, is that if you also just have patterns of planar growth, you can have regions of stress buildup, which can also define where folds form. And so this is actually in this paper, which again, if you want, we can talk about it during coffee break, lunchtime, or whatever. But basically what we showed is that if you have also differential growth in the plane, you can sort of extrapolate how, from that simulation I showed, if it's in 3D, you can get folds in certain position, okay? For example, here. So this is kind of a summary. So in this story, what we did is to generate this 3D finite element model, where if we have differential growth as we measure in the tissue, we end up generating 3 folds like what we see in the animal. But if you had uniform growth, all else being equal, okay, then you get uniform ripples. So this was the kind of the paper that showed that, in fact, growth in the plane, because of the stresses it generates in 2D, actually translates into 3D deformations and induces folding and buckling. Okay. So yeah, so I think that's a very, very quick whirlwind of the whole kind of mechanobiology for anyone who's not heard of some of the sort of intricacies of how the biologists have really worked with the physicists to try to merge the field. Okay. And before we go on, are there any questions here, or do we want to take a quick 2-minute break before we go into case study? No? Yes? No questions? Yes, yes. Yeah. Okay, so those hexagonal things are actually are collectives of cells. So these are individual, what they call omatidium. And within each omatidium, there's actually, I don't know, I can't remember the exact number, but like tens of cells, 50 cells or so. And then they collectively form this perfect kind of compound eye of the fly. And the reason why this was used as a genetic sort of model or a model for biologists, because normally the eye is so perfectly latticed, like a crystal lattice, right? So because of the precise cell number, cell patterning, cell shape, cell packing, it gives you this almost like a glossy, smooth eye, which the fly needs to see perfectly, right? But geneticists found that if you have tiny mutations, for example, in the Hippo pathway or EGFR pathway, Wnt, all these pathways people know causes growth defects, cancer, they were actually first discovered in the eye of the fly because it was so sensitive to any different modifications. Even if you had one single cell growing a bit too much or dying a bit too much, then the eye becomes no longer smooth. And so this was a very good readout to carry out, for example, genetic screens that people have used to find, for example, the Hippo pathway that Yorkie was found in the screen, where they show that, if you mutate this pathway, you get rough eyes. So that's kind of the reason and the history behind that. Yeah, so it's actually, so as biologists, if you want to, for example, study hundreds and thousands of genes, right? You need a system where you can study the effects of, for example, mutating these genes one by one, and yeah. Mm-hmm. Yes. Yes. So yes. So I think you're asking. So what I showed was the final output, right? Absolutely. So what you can do, and I'll show you in this case study, is that you can essentially, let's say you look at the adult and you go, oh, there's a problem. But where did that problem, or when did that problem occur, right? So you can go backwards in time. So you can look at the animal, let's say 10 hours earlier, 40 hours earlier, 7 days earlier, whatever, and exactly track when did that problem occur, because that's key to understanding the whole reason behind, or the mechanism behind that error, right? Yeah. And then from that, you can say, okay, maybe it was, maybe at stage, I don't know, X or day 2, cells were growing so much more in this mutant, and then that kind of accumulated over time. But then maybe afterwards, actually, it was perfectly fine again. So again, as developmental biologists, what we want to do is understand these developmental defects and when do they occur, when is the most critical window. Because if you want to, let's say, treat developmental diseases or treat errors, you probably need to know when, where, how, right? These are the the usual obvious questions that we'd like to understand. Yeah. Okay, cool. Yes. Yeah. Mm-hmm. Yeah, so this was just something we forced in, right? So this is obviously nothing is ever purely symmetrical, but for the case for as every model, you have to simplify it to some degree. So in this case, we were more interested in the position of the folds, and the weighing to near proximity is very symmetrical, kind of top and bottom. It's not perfectly symmetrical, but it was symmetrical enough that we just simplified it. So we just treat it as a top bottom symmetrical object in this case. Yeah. So this is purely a simplification of your system. As always, models are simplifications of a system. Yeah. Yes.
right?
Mhm.
One population that's growing and the rest.
No, so again, so we could use this as a case study tomorrow if people are interested in it. So essentially what we did is, in this simulation, we measured and we showed that actually there were these stripes of differential growth patterns here, which is what drives these, but we also did artificial growth modification experiments where, for example, we made patches of cells that grow faster than normal. And as a result, you often see like at the boundaries, there will be like a fold or a buckle to cope with that differential. Basically, if two populations have to, grow differently, how do you accommodate for that? And often a fold is the way. Yes, so we did various modifications and perturbations, but as well as, in this case, monitoring what the real growth differentials are and putting those into the model to see if that was sufficient to give us these changes that we see. Yeah.
Okay.
But yeah, so these are great questions, which maybe we will kind of yeah. So if people are interested, tomorrow we could spend a bit of time going through this paper in a bit more detail. But again, we can talk about it at coffee or lunch. Okay. Right. Should we move on then to? So yeah, half an hour should be all right. Sorry, as I said, I'm incredibly dehydrated, so I need to keep topping up on water. Good. So again, I always like to use this phrase, and I'm sure as physicists, you're not sort of surprised maybe if you've worked with biologists when they always come and say, look, but your model has missed this, or you didn't capture this, or what about this process? You didn't add it in, so how can your model be correct, and so on. I don't know if any of you have faced this feedback, criticism from your collaborators. I certainly did when I started to, build models, and it was very much, I don't know, a constant debate, fight, argument with the biologists. And I think this is why I always use this slide, right? All models, it could be a mathematical model, a physical model. It could also be a model of your whatever animal system, right? Cell culture is a model, fly is a model of, whatever humans, as we like to think. So everything is a simplification, and you have to just, I guess, decide on how much to simplify, right? What is sufficient? What is good enough? When is a model good enough? Of course, it's never going to be perfect, and so then hopefully it can be also useful for whatever system you're trying to answer. So, I mean, I don't know. I just wanted to, again, maybe this is preaching to the choir, but again, I found kind of when I work with. So anyway, I'm often stuck in the middle. So we work with pure biologists sometimes with the models that we create, but also as experimental biologists, we also work a lot with theorists. So I find that depending on who I'm talking to, I often have to adapt the way I communicate, the way we sort of discuss things. And certainly, I think most of you here are theoretical physicists and so on. Your biggest challenge is probably how do you communicate with biologists, or they may come to you and say, look, oh, I have a question. Can you just model it for me? And then you're like, okay. But I don't know if you had this. So I don't know. So I felt like they biologists often think of models as either completely magic, as in we can do whatever they ask us to do, or they completely do not believe it at all. They just think because you can model whatever they want, they also don't believe it, right? So I don't know. I find it's being quite a challenge for me to get them to kind of appreciate it in the right way, agree with it, but also not completely think it's either magic or rubbish. So it's like, how do you get the middle ground? Okay, and so this was one of the first problems I worked on as a kind of I don't know, biophysicist, I suppose, during my postdoc, and I found this example was always a really good example, whether the audience is sort of more theory or more experimentalist, to really see how to kind of merge the middle ground, and hopefully you'll kind of appreciate that. And so what I often teach, whether it's mostly to biologists, is that how do you actually use models in the kind of the right way? Or in this case, and if you're working with biologists, how do you convince them that your model can be useful and in fact incredibly insightful, right? And often the issue is, well, what is your question? It seems obvious. What is your question? But I also often find that the biologists always come with very vague questions. For example, it could be, oh, I just want to understand how my cell divides, or how it grows, or and you're like, how am I supposed to? Yeah, exactly, right. So I see the results, I was like, okay, sure, but how I can't tell you. Anyway, so I guess the point is, biology is obviously very complicated, And generally they come with 1 million experiments, 1 million data that they did. And you're like, okay, can we simplify this? You kind of have to force them to simplify the question, right? And essentially it means, can you frame it more precisely? So for example, yeah, like how does an organ get a shape? You're like, okay, so great question. But what about, so can you frame it more precisely? And if possible, can you actually sort of force them to come up with a more concrete hypothesis, right? Again, basically, can they be, or can you be as precise as possible? And this will help them, help you, help everyone. But often, even just these few steps takes, I don't know, weeks, months to kind of define precisely. And often at this stage, what we find with our collaborators, they disappear off for years. and honestly, they're gone for years because they realize, oh, actually, yeah, I'm nowhere even near coming up with a model or helping you with a model, because they really haven't a clue, how to formulate their questions precisely enough, and so on and so forth. Okay. But if you do eventually get to this stage, hopefully then you can really communicate with them and say, look, okay, great. If you have a concrete hypothesis, what do you have? Hopefully you have experiments, therefore, to support this. What are they? Can you explain them to me? And then maybe we can generate a simple model to test this hypothesis together, right? And it should be a constant kind of iterative process. You should be communicating with these biologists in day to day. So by the way, can I just ask, how many of you actually have worked closely with, let's say, experimental biologists? Okay, great. Okay, cool, good. That's good. Good to know. So again, you might sort of have experiences and resonate with, or you might also, it would be nice to hear about how your experiences have been as well, and which parts worked well, which parts didn't work well, and whether, where the biggest barriers were, so to speak. And so did you find often the first few steps was the biggest challenge? I don't know, it would be nice to hear some of your thoughts. And what was the biggest challenge in working with biologists then for all of you who have? Yeah. Yes. Yes. Yeah, exactly. So these, again, I sort of split them into these steps, right? Again, they're not chronological, although I find most of the time, yeah, kind of working through these steps does help. And communication, exactly, and just finding the same language, because they could be talking about, oh, I know genes ABC affect XYZ, but then they want to understand it from the point of view of physical models. Okay, so what do these ABC genes do to the forces? How do they work? It's like trying to link it all together. Yeah, cool. Any other kind of major kind of, I don't know, obstacles you found or challenges you found? Yeah. Yeah. Yes. Yeah, definitely. Exactly. So anyway, so let me just kind of, and I think, yeah, I mean, this is great, because talking about it, at least you've all worked with, well, most of you worked with, hopefully some kind of concrete model and some kind of concrete problem. So you probably understand this. But I find, again, if I'm often talking to this, let's say, to biologists who've never tried to even build a model, it can almost sound too abstract, just this, they just can't even appreciate these, right? So I like to use this kind of next few slides as a problem, just to illustrate some of these concepts, all right? So yeah, bear with me, we'll kind of go through this fairly quickly. So this, as I said, this case study is really about the concept of how do you orient divisions, okay? And as a result, as we talked about earlier, these division orientations will also control the overall shape of the wing in flies, okay? And so I guess the biggest question really, let's say the biologist came and said, oh, I want to understand how organ shape is controlled. So you're like, great, yeah, me too. I'd love to understand, but which organ do you want to use as your model system? Even already, that's simplification. And so in this case, we used the fly wing, okay? And the reason for that is because like the eye, as you mentioned earlier, the wing is a great model tissue to look at shape control. And the reason for that is because in normal wild type wings, almost every wing is exactly the same size and shape, okay? So it's what we call really robust system. And so if there are little variations, then it's very easy to sort of see them, but then understand, where they came from, okay? So it's therefore a great system to understand how do you control organ shape. So already you can see that's a simplification, right? Because we went from this big question, how do we shape organs, to, how do we shape the wing in the fly? So that's already a step. And then, as we were saying earlier, this is already the final outcome. So almost it's too late, right? I mean, you can see a problem. So you have to go back in developmental time to understand where did that wing kind of come from? What were the processes that led to the wing? What are the key, so this obviously requires, hopefully the biologists have worked with their system for a few years, so understands the system a little bit. And so in this case, I don't know if I'm sure most of you have heard about the Drosophila wing as a model system. Yes, no? Who's never heard of? Okay, good. Okay, that's good. Okay, so I'll quickly therefore introduce that to you. So basically this adult wing, okay, takes about 10 days to go from an embryo to that adult wing. And there are a few key steps. So this step here from 50 cells or so, the cells are undergoing rapid growth, division, okay? And in the end, you get this 50,000 cell tissue. And so this is really the key sort of 4 days of growth and morphogenesis that the tissue is undergoing. And pretty much the size and shape at this stage, called the end of the wing disc stage, sets the shape of the adult wing, okay, for most purposes. And so therefore, if you want to understand how this is controlled, you really have to understand how this process of development is controlled, okay? And as I said, you can also see the wing folds. So people have also used it to understand folding, for example, as you mentioned earlier, okay? But for this problem, let's stick with this 2D structure, basically a patch of tissue, okay? And so then again, what's good about the wing as a model to look at shape is because there is this stereotypical shape, okay? So the wing generally has this elongation, this oval shape, which is elongated. So I'm going to have to, unfortunately, use a bit of jargon here, which I hate. But again, biologists love jargon, so you have to kind of cope with that. But essentially, what's called the proximal distal axis. So proximal, very simple, close to the animal. Distal, far from the animal. And this axis, proximal distal axis, sets the kind of the long axis of the wing, okay? And however, to make things a little bit complicated, as I said, by this stage, if you look at the clone of the wing, you can see these cells kind of grow and divide in this long axis. But during the wing disc growth, this axis is actually a radial axis, okay? So what happens is that basically this middle part, as the animal develops, will pop out of the screen, will jump out, okay? So then this part becomes this distal far part, okay? And this proximal ring. So imagine this kind of comes out like a sock, kind of extruding out of your screen, right? So then, can you imagine that? I don't know. I should have brought a model, but anyway, imagine the screen kind of pushes out, okay? So then this outer ring is this bit, okay? But simplifying this, again, simplifying the problem, basically this distal axis is this kind of radial axis, okay? And the reason why I'm spending a bit of time to look at that is because remember the question, how do you actually control divisions in the wing, right? So again, you have to surely pick a system where the division orientations are somehow controlled, because if it's all random, there's not much control, then maybe it's not a great system to study division orientations, right? And so again, biologists knew that in this tissue, if they were to do this experiment called clonal analysis or lineage tracing, which is essentially you label the cell, let's say with a color, the GFP, and then all of the daughter and granddaughter progeny cells will inherit that color. And this allows biologists to be able to follow the lineage of a cell, right? Because if you look at all the cells at once, there's no way to know how this final pattern emerged, right? And so what biologists have done is you can label a few sparsely, let's say very young, you label lots of single cells, and then you let these single cells grow, and then you can trace this pattern, okay? So then this clone here, okay, basically came from a single cell. And from this, you know that this sort of final emergent shape of the clone was because of one single original mother cell, right? and it divided, it grew, it slightly, I don't know, underwent whatever shape changes and so on to give you this final shape. Okay, does that make sense? Yeah, because if you didn't have this, there's no way to know where this whole, all these thousands of cells came from, right? Yeah. Yes, it could be. Yes. So that's a great question. So what again? For example, this might be a few that's kind of split off, right? So these are things you could account for and so on. But again, you can design experiments where, or for example, these little ones. So you can sort of fine tune this. You could, for example, image for T1 transitions. What's the T1 transition frequency? You can put all these into the models if you want to. So we can go into that later. Although, yes, yeah, go on. No. No. Okay. It's quite technical. I can go into if you want. So, okay. So, but quickly, what we do is these are. That's why Drosophila is really amazing. And as one learns more about these, basically we have these constructs that all the cells have. Okay, but they also have what's called heat shock inducible. And so it's probability based. So let's say you have 100 cells, you put them in for 1 minute at 37 degrees. Stochastically, let's say 5 of them will pop. And that's all. Yeah. And the details we can go into. Yeah, exactly. Yeah. So you can sort of, by tuning that time, so let's say 1 minute, 10 minutes, 20 minutes, I can set the random density. And most of the time, there will be sparse, because there's no reason why they would cluster. It's kind of, yeah. Yes. Oh, yeah. Oh. The green and red. Okay, yeah, sorry. So, so the green here is probably nuclear staining for DAPI, just to be able to label all the cell nuclei. And the red is probably actin or myosin here, just to label all the cell outlines, essentially. Yeah. So there are, again, many markers we will go into a little bit that people can use to label the cells so you can see the cells. Yeah. Okay. Right. Good. So think of it as these are all ways in which biologists can do experiments to generate the parameters you need for your models. Right. This one? Yeah. Yes, it is. Yeah. So the wing is 2 layers with basically not much in between. But exactly. So again, I always say I should bring a sock, but essentially, yeah. So if this comes out like this, right, it's flat, and it comes out like that. Yeah, exactly. And so that's why, again, I digress, but there are some wings that have blisters in them. That's because the 2 layers didn't manage to oppose correctly. And actually people knew this was because of adhesion. So let's say you've probably all heard of ECM, integrins adhesion. So classically, these bubbles between the wing were because of integrin mutations and they couldn't adhere. Yeah.
From a single layer of cells, or are they from a single layer?
So when they're at this stage, this is a single layer. But imagine this single layer, when it comes up and domes up, these 2 single layers then become 2 layers. Yeah, exactly. Yeah. So for simplicity, we think of this at this stage as a monolayer, as a single layer of cells. Yeah. Okay. So again, simplifying the problem, single layer easier than multiple whatever layers. Okay. Yeah. So there's unfortunately there's a little bit of geometry to kind of appreciate the problem. Anyway, so exactly. So in this view, most of the data will be in this view. Think about a single layer of cells. So we've gone now from basically it's this view. Okay, you zoomed in. Okay, yeah, and then so now we're looking down, top down. So these are cells that spontaneously, randomly, because of the heat shock, expressed GFP. So let's say 5, well, I don't know how many, 10 of them expressed it, and then 10 of them were allowed to grow for about 2 days or 3 days, and then you follow their patterns, okay? And so from this, at least we know, okay, something, whether it's cell division orientation, whether it's intercalations, whether it's all these processes I described, in the last half an hour, something has caused this patch to grow in this elongated way, right? So how has that occurred? How does that happen? And so then the question is, does your collaborator or biologist have a more precise hypothesis, right? Then, so already this is pretty good, right? You force them to say, look, I want to look at organ growth, I will look at wing growth, wing shape, clone shape. And so now they say, I know clone shape is this. How does that clone get to this shape? That's already pretty good simplification and formulation of that problem, right? And so then let's say you come in and say, okay, do you have some hypothesis? Any rough, vague idea? Does that have anything to do with mechanics, or are you talking to the wrong collaborator here? And then they go, actually, yes. I saw this really interesting gene called DAX, but it's actually a kind of myosin. And then as mechanics, you hear about myosin, you go, oh, wow, that's going to be interesting. It's going to do with mechanics. And they tell you, oh, this weird myosin is expressed in this really kind of cool polarized way. So like this here, basically only one kind of side of every cell has this myosin there. The other sides, so let's say a quarter of every cell has the myosin. The other side doesn't have this myosin. What's going on, right? And their hypothesis could be maybe this polarization polarizes the force, which polarizes the shape, which polarizes the division orientation of these cells, right? So can you see already you forced them to kind of come up with a bit more of a concrete hypothesis and almost like a conceptual logical pipeline to how it could work, right? And then you're like, oh, actually, that's interesting that we could work with this, right? And so, but of course, you need to ask them, okay, can you do one killer experiment for me? Which is, can you show me this hypothesis that we have? Actually, this DAX gene, does it even have a role in division orientations before I go and spend, a year or whatever developing a model for you, right? And so what kind of, what would you need? What data do you need from them before you're convinced? So let's say this is a hypothesis. They hypothesize that DAX, by being in this weird polarized pattern, does something to these cells somehow and affects their division orientation, right? And you either believe them or you want to see some data from them before you believe them. Like, what would you want? What do you want from them? As a very, not like detailed, just a crude. Exactly, right? Hey, you're not allowed to, you're not allowed to answer. Did you hear him? Yeah, you heard him, right? Yeah, exactly. Knock it. But exactly, knock it out. Yeah, exactly, knock it out, right? So no brainer. What biologists love to just knock things out and see what happens. That's how most of the things we do are kind of done. Exactly. So they need to do a whole bunch of quantifications, ideally, right? Not just these pretty pictures. I want some numbers. I want some stats, right? So this is the control or the wild type. And indeed, if you quantify these clones, right, orient them, again, I don't want to go into the details, but essentially if you quantify them, you'll see there's a bias in the sense that most of them are oriented along this theoretical radial PD axis, okay? And so if you quantify hundreds of these clones, you'll get a map that looks like this, all right? But if you knock out the DAX, or even cooler, if you overexpress the DAX everywhere, i.e. if you kill the polarity, then you kill the normal patterns of the clones, right? So pretty obvious the difference, right? So normally there's a bias. Most of them have this theoretical radial orientation, okay? But if you remove the polarity, either knocking out or overexpressing, then the clones have no directionality. They are now naive to this polarization of direction. So I think then now I'd be convinced that no matter how, but somehow this DAX gene seems to be controlling division orientation, which makes me want to then maybe do a model to help the biologists understand. Yeah, Billy, sorry. I don't have, but they look rounder. Yes, exactly. So that's actually the very first piece of data that we got from the biologists. The adult wings were just cute little round blobs. Yeah, exactly. But that could also be many reasons. It may not be because of the clones. So this was really the first data to show it's really because of division orientation. It could have been something much later on. There's a million processes that kind of affects the final shape of the wing. Yes, but they are now linked. Yeah, exactly. Right? Did you have a question? Okay, yeah, exactly. So great question, because again, before then we were like, so what, right? Like, yeah, good. Okay, and so then you're like, okay, great. I'm kind of convinced, right? So let's come up with some ideas or hypotheses. It could be, for example, like I said, it's a myosin. So where it's located maybe it generates more contractile force. Not surprising for myosin. And as a result of the shape polarity, the divisions will be polarized. Okay? So remember, we said that cell shape really drives division orientation. And so if DAX affects shape, then it's very likely to affect division orientation. And that's probably why these clones are elongated, and that's why the wing is elongated. Okay? But it could also be another counter hypothesis. So again, I really like it when the biologists also have another hypothesis that they just can't, they don't believe in it, but they also can't negate it because, you know, you always have your favorites, but again, you should be open-minded if you can, right? And so we kind of said, okay, well, but what if it's, for example, has nothing to do with forces, nothing to do with shape, doesn't affect the shape of the cell, but maybe it binds spindle. There are lots of molecules that tether spindles, that bind spindles, and this could bias the spindle orientation and also contribute to cell division orientation, right? That would have been equally valid hypothesis. So tell me why you prefer this one to that one. And they were like, no reason really, just you know, an idea we had. So okay, so we're like, okay, well let's try to keep these both in the race and see which one basically is the final solution, right? And so we were very open-minded and we kept, you know, both hypotheses kind of together. And so again, basically we kind of went through these steps, okay? And so now we were, okay, are there experiments to support one of these or, you know, negate the other one, right? And so we wanted to see a bit more. And so then I ask you, what would you then do? What's the fundamental difference between these two hypotheses? Yeah. Yeah. Exactly. So as I've drawn, for this to work, there has to be some elongation of the cell prior to mitosis, and hopefully that's correlated with this DAX polarization for this to work. This doesn't care about shape, just does it, right? Exactly. And so then this is where we really, basically this is exactly what you just described, okay? For this to work, you have to have some oriented growth of the cells before the division kicks in, right? For this the other hypothesis, this is not a requirement. And so then this really was a problem for bio, which is actually me in the case at the time. I had to generate supporting data for this. And so this required some live imaging. Again, not new now. Lots of data you get, I'm sure, from your collaborators are live imaging, right? Cells undergoing morphogenesis, shape changes, whatever. And so this is a movie where, I don't know if you can see, maybe you can see in these bigger ones, that this is a cell undergoing mitosis, dividing into two, yeah? Maybe not, but anyway, these are some stills. So essentially what we could do is to actually take live imaging. So this really required live imaging data, not just stills, because you cannot infer what the shape was, 5 minutes before and 10 minutes later from fixed images, right? And so then, and this has really revolutionized, I think, a lot of the data we have nowadays, right? Not just the dynamics, but even just for this. What is the shape of the cell just before it undergoes cell division? Is it elongated? And pretty much always the cells have this transient elongation phase before they divide into two. And there's a beautiful correlation between the division axis and the basically the shape of the mother cell and the division axis, okay? And so this again proves that in our system, this Hertwig's rule that cells orient and then divide down long axis also works. And so we'll sort of skip this little detail. So essentially what we've kind of shown or forced our collaborators to show is that indeed the cell shape is what's driving division orientation, okay? Yeah. Say again. So I mean, there's obviously, as we said, there's if we quantify 90, whatever, 100 cell divisions, and yes, there's a range, right? So not all of them will grow to perfectly elliptical before the divide. There's reasons, many reasons for why that is the case, but in general, the average is they will orient and divide. So for example, this is showing you this, right? So some of them are not very oriented, but we'll go into that later. Okay? So I'm just a bit conscious of time. So anyway, so basically what we've shown is that it looks like more likely this, right? This hypothesis on this side. But then the next question is, well, does DAX actually control cell shape? Because again, if DAX doesn't control cell shape, this is, wrong. And so then the next experiment is to actually test that. So again, what biologists can do is, if you want to know what DAX does, what do? Knock it out, right? So we knocked out DAX and realized that actually, if you remove DAX from a cell, the cells become more enlarged. And the vice versa, opposite. If you overexpress DAX everywhere, the cells, in this case, I haven't drawn it, but basically this is a condition where DAX is everywhere, the cells become more constricted. So what this shows is that at least mechanically, this molecule of DAX, which seems to look like a myosin, actually does have contractile ability. And so then we also did laser ablations to measure this contractile ability of DAX. Okay, so the exercise I'm setting you later actually is about laser ablation analysis. I don't know if you've done any of that yet in the last few days. Okay, good. So basically, this is a technique that has really been critical for biologists. You know, how do we measure these forces? This tension we keep putting into the models, right? Where do they come from? Where do we measure them? And so this is a technique where essentially you can model it, for example, as a Kelvin Voigt model. So like a viscoelastic material. And the idea is that if you have, let's say, this elastic band under tension, right? If you cut it, the higher the tension is under, if you cut it, the more recoil you would expect to see, right? But you can actually model this much more precisely than what I've just discussed. And this is actually the exercise I've set you. So we can go through this a bit more. There will be, I don't know if have a think about it first. On Wednesday, we can talk through this in more detail, because I guess now it's not the time for that. But essentially, a very rough kind of relative quantity we can get is if you cut a very tense junction, tensile junction, the initial recoil velocity is proportional to the initial force that this junction is under. In fact, it's proportional to the force divided by the viscosity. So we can go through this, later in the exercises, okay? By kind of solving this, or in fact, just fitting this equation. So if you want, we can share this and take a note of this. But anyway, the point here is that what we showed is that if you have DAX present, these junctions are under more tension when you ablate them because of this high initial recoil velocity. On the junctions where there's no DAX, the recoil velocity is lower, okay? So again, this is linking to this hypothesis then, that in fact, this might be true, because where there's DAX, there's more measurable higher tension, okay? And where there's no DAX, the junction is floppier. Okay, does that make sense? And so then by now you can sort of conclude that actually it sounds like this DAX polarization for sure is affecting shape somehow, affecting spindle orientation somehow. It could be still doing it directly, but it also looks like it's doing it via controlling cell shape in a polarized way, right? And then you have to ask, okay, well, there's lots of things that can affect cell shape, right? So a bit like what you were saying, it's not just the forces of DAX. Sometimes they look rounder, sometimes it looks less round, because there are neighboring forces, neighboring cells, that will also feed into and merge with the forces of DAX to affect cell shape, right? And so what are these other forces? And in a way, this is when you say, okay, maybe the vertex model is the right model. So again, I use this because you've all heard about the vertex model now from last week's and the few weeks lectures, right? And so this is a model where I won't go into this. I think Lisa spent quite a lot of time looking at this. So it's a model where people have used to analyze, to try to basically model the forces just in one apical surface. So simplifying the problem into 2D, which is what we're doing, to understand the force dynamics. And I will skip through this because you've heard about this, hopefully, and remember. The key terms are area terms, the line tension term, which is a combination of contractile forces versus adhesion forces, right? Do we remember? Do we nod? Yes? No? Okay, good. And then there's also the perimeter force. And so hopefully we can skip this because we don't need that. And so then the idea is, how do we include this DAX force into the standard vertex model, right? So we've established that DAX increases junctional tension on the junctions that's localized then. And so which term would you use, which term would you modify if you wanted to include the forces of DAX in the vertex model? Do we remember? So not the highlighted one, sorry, but which of these terms would DAX fit into, right? So this is basically, you start to parameterize your model, right? So all the experiments leads into the stack's doing something. So as the physicist, you're like, okay, this is where the biology probably needs you, right? Where do I put it? Like which term, right? And so what do you tell them? Yeah, okay, yeah, go on. We're running out of time. Exactly, second one, right? Because we said the second one is the term where is kind of predominantly balanced between contractile forces between two junctions and the adhesion forces, right? So if you remember this term, if it's very high value, it represents very high contractile force. And so that junction decreases in length in the simulations, right? And the opposite, if it's very low or in fact negative, you can have negative values for this, then the red arrow dominates. So basically it simulates if you have two cells that are sticking together, the interface will grow if there's more adhesion. And so DAX most likely fits into this term, the blue term, because we've measured that it has higher tension, so it should contract more, right? Where it's localized. Does that make sense? Yeah, exactly. Good. And so, in fact, what we did is to do a trick, essentially that. But remember, the complication is DAX is polarized. It's not like every junction has more DAX. So where do you put it in? You can't put it into every junction. So what we did is actually a kind of a trick where we use the sine function to I mean, there's many ways to model this. Of course, you can say, look, depending on the angle, we add a, weighting. If it's perfectly perpendicular, you have 1. If it's horizontal, you have 0. But basically, the sine function does this, right? So if you look into the details of the maths, you'll realize that essentially on junctions this way, you have very high DAX, this way, 0, and then in between. So this takes into the idea of polarization. So basically, you can polarize your tissue so that all the forces are not equal. And then you look at the effect of what's going on there, okay? And that, as I said, there are other ways, of course. And so just to show you the final kind of few slides. So this is essentially a control simulation, or in fact, it's simulating the DAX mutant, right? Because we don't have any polarity. Everything is the same. Everything is equal. However, there's still one major rule, which is the cells, when they're about to divide, they measure their longest axis, and then they divide to cut the long axis. Because that is a universal rule that seems to be the case. And hopefully you'll see that, what do you get? Basically a circular isotropic growing tissue. Yeah? Which one? Sorry. Oh, yeah. So the growth is the same, okay? So the growth, so in that one, it's because we had more growth in the middle, right? And then less growth. So there was yes, this is how many, yes. So that actually was a follow-up of this, but anyway, yeah, exactly. So this is same division everywhere in terms of density and range, yeah, okay? So it's only about the force polarity. But then in this simulation, we are now essentially simulating the wild type situation where we have polarized DAX, okay? And we just have a little bit of increase in tension on the more vertical junctions, like in this cartoon here. And hopefully you'll see, or else equal. So everything is exactly the same as the previous simulation. And what do we see? Most people said a worm. Most people would say a worm at this stage. Most people, oh, that looks like a C. elegans worm. Something elongated clone, let's say, right? I'm not claiming it's a wing, but yes, a wing, a long structure, right? And so, and what I found quite amazing was, it's not as if all the cells are being stretched, right? It's very subtle, right? Because the polarity is actually quite weak, but just enough to bias the divisions so that the emergent behavior is very different. I mean, there's no denying the whole shape of the collective epithelium is dramatically different. But if you look at every single cell, it's not that obviously different, right? But the computer obviously can sense that, can measure that, okay? So, and then remember the other hypothesis that was still in there? Like, what if it sort of tethers the spindle, forces the spindle to divide in this perfect PD axis? And what happens is kind of a B's on it, because it's so perfect. Like the spindle, if it's forced to be PD, every cell division is perfectly PD. Whereas the cell shape mechanism gives you a natural noise, right? A natural variation to this imperfection, which is actually what the wing is. It's not a single cell long tissue, right? There's a width to the tissue, okay? And so I think we were convinced that this mechanism doesn't seem to work in the system. So of course, as always, I'm sure you've all done this. You run your simulation, a million times, and you plot some graphs, and you try to match it to the experimental data, and so on and so forth. And so, yes, we kind of convinced our collaborators that probably, most likely, DAX does do this via this tensile mechanism, changing shape. Yes. Say again, sorry. The elongation ratio, yes, sorry, that's of the clone. Sorry. So this elongation ratio is just if we take the clone and just take the long axis divided by the short axis of the clone. Yeah. Just because essentially that means, in the spindle mechanism is so perfect, it's very long and thin, right? Whereas if you have, let's say the DAX is elongated, but it's not perfectly elongated. So this requires, obviously there are details. So this requires a little bit of parameterization of this DAX factor that you put into the model, right? Because if you have it super strong, then you also force every cell to be like bricks, and then everything will be perfectly. So there is obviously a bit of, as always, setting this new parameter. What is that new parameter? How should you put it in? And this is where we got a lot of criticism, right? You could just say, well, yeah, skeptics saying, well, you just tweak your parameters. You can always give me what I want, right? And what does that mean? How does that even help you? Like, convince me. You didn't just tweak a parameter. And it's true. I could also have made this look a lot worse, right? And so if you're okay, one last slide just to convince you that no matter how I tweak the parameters, I could never get it to work if it's not this mechanism, okay? And basically I could, it will never look wild type, even no matter how I tweak. And the reason is that this I really love, because all of these require simulations and so on and so forth, whereas this analysis really convinced the skeptics with actually pen and paper analysis. It didn't require us to do simulations. And the reason, I'll just bear with me, is basically we call this delta analysis, okay? And the delta by definition is, imagine you have a mother cell and you have two daughter cells, right? And you calculate the difference in angle between their alignments. There's always a bit of error because there's never kind of perfect division. And so then you define delta to be one direction, doesn't really matter, but basically you calculate the difference. So sometimes it can be negative delta because it's pulling the cells more towards PD. Sometimes it could be positive delta, i.e. the other way. Okay, does that make sense? The direction is just, you define it, right? So if the cells are reading the shape of the mother cell, and then sort of measuring that, dividing, you could imagine it could have errors equally on either side. There's no reason why it should either go left or right more, right? And so the delta, if you were to plot the delta, it should be kind of a directional distribution, right? But remember the other hypothesis that if somehow DAX is at the PD axis and forcing the cells always to bind and correct itself to this axis, then this delta analysis would always be more towards the PD axis, right? Because it's physically correcting it every time. And so if you were to then plot a graph of delta, you would expect always one direction, okay? So then essentially we take our movies, pen and paper, whatever, calculate this delta and plot it. Okay, and this is what we got. So elongation ratio means what is the elongation ratio of the original mother cell, okay? And this delta is what I just explained. And what we saw is what we call this Christmas tree distribution, which is kind of amazing. So very clearly you have is symmetrical, right? But also what we didn't quite think conceptually is that if the longer the cell, the tighter or smaller the error. And that makes sense. So you spotted really well that you also have some really small, not so rounded, not so elongated cells. They divide, but with much more error. Because a round cell, well, I don't know what Milan, I just sort of tried to divide, right? But the longer you are, so if you have a really long cell, they will always divide with very less kind of error. Then we did the same analysis on our simulations. Okay, so this is with one set of parameter, but we can change this parameter. We can have much more noise. And so with the DAX affecting shape model, we also get this similar qualitative distribution. which is you get negative and positive delta values with equal probability. But if you simulated this other counter hypothesis, which is they force the correction, then we always get a bias in one direction. And again, you can have much more spindle wobble, you can have lots of error, you can tweak parameters that will only affect the kind of the quantitative bias, okay, but never the qualitative shape of the distribution. And so this was, for us anyway, the really killer that kind of killed all skepticism in this model, okay? And so I love kind of describing this because I think this really doesn't need sophisticated, fancy simulations or huge computational power, but just a little bit of thinking actually really helped us. Okay? Hopefully you're convinced. So maybe we can talk about this at the end of tomorrow's session since we're a bit, or yeah, okay? Yeah, but also let's do that. Okay, cool. Right. Sure, fine.